Dum dum. Good morning, everybody. Oh, hang on, wrong one. Ah, beat me to it. All right. Get my brain working. So what we're going to do today is finish up translation. And actually, I want to go back over and just show you a few things about elongation, which we kind of skipped over a little bit last class before we move further on. So, ding, ding. Get rid of that. Where is it? Aha, that's the one. So basically, we ended up. Did we get to termination yet? I know we did initiation up the wazoo, but I can't remember if we covered termination or not. Anyone remember? Oh, there's chat. If I can get to the damn chat. No. Oh, thank you, Sophia. Appreciate that. Okay, so essentially we've covered transcription, translation, translation initiation. And one thing that I want to just reiterate again is that the key differences between prokaryotes and eukaryotes. Prokaryotes, it all assembles on the start codon using the small ribosome, right? So the Shine-Delgado sequence is the key there, whereas in prokaryote and eukaryotes, sorry, my brain's all kind of fuzzy. Spent a long time working on the car yesterday, so I'm kind of pooped. It's a scanning mechanism. So the whole complex assembles over the 5' cap using these initiation factor cap binding proteins and some help from the 3' UTR and the poly A binding proteins. And then that whole complex scans along the 5' UTR until the methionine RNA that's in the P site, well, it's not in the P site, it's attached to the small subunit, until that finds an AUG, essentially like a barcode scanner. And then all of this stuff assembles. So even though the components are largely the same, the mechanism by which translation is initiated is quite different. What do you want? Come on, up you get then. Up. Come on, sorry, I've got to get my foster pup up on the couch. Come here. There you go. You did it. Good job. I don't know if you've seen her. Hang on. There's Lilybit and my foot. Hello, foot. Oh, that's one. Here we go. Being a little pest, aren't you? Hopefully she doesn't start chewing on stuff. Anyway, what? No, I'm not going to play with you. Okay. All right. So once that all starts, right, once a start codon is found, either by direct binding in the case of prokaryotes or by scanning in the case of eukaryotes, then we're off at the races, right? You know, now the whole process begins. And elongation is really a series of repeated stepwise processes. So the first thing that happens is another tRNA comes into the A site. This is helped by a couple of proteins, or a protein. So I'm just going to write this down for you. So EF is elongation factor rather than IF, which is initiation factor. So EFTU and GTP are used to bring in the next tRNA. And that's kind of a similar, essentially the same thing happens in eukaryotes, other than the fact that the names are different, essentially. And so GTP again plays a role. So if you remember, GTP is also involved in assembly of the T-met RNA onto the start codon in prokaryotes. I'm not actually 100% sure in whether GTP is used in assembly in eukaryotes. I don't think so. Anyway, so same molecule, and again, it's the analogous form of ATP, right? So it's not ATP because it's got a G in it, but it does the same purpose. It's small, usable energy. So this complex forms, right? So this is the video that's linked in just up here. And I'll put that, actually it'd be easy, just as easy to put this in the notes. And it's worth watching. It's a pretty neat video. Very straightforward. Where's the chat box? Here's the chat box. There you go. Come on. All right, so essentially this all forms complex of the new tRNA, right? So presumably these are kind of hanging around as complexes anyway, and they're basically waiting for their codon to come up, right? So you can imagine there's this big cloud of tRNAs complex with EFTU, right, which is the kind of the axis elongation factor, and GTP, all kind of hanging around, right? And so when their codon comes up, those then enter, and in the process, GTP gets hydrolyzed to GDP, right? So the energy in that phosphate bond is what powers this process. Go on. Hang on. There you go. So essentially then, so again, you can see the purpose of the anticodon, right? That's on the tRNA that recognizes the UCC in the RNA because it has the exact opposite on the anticodon, on the tRNA. So once these are together, now we have a tRNA in the P site, the peptidyl transferase site, and a tRNA in the A site, the access site, both with amino acids attached to them. And so the very first step is to cut this first amino acid off, right? And that's carried out by the ribosome, if you remember the catalytic RNA in the peptidyl transferase. I'm just going to skip ahead. Yeah. So essentially what happens first is that amino acid gets cut off, and this is going to repeat over and over and over and over again. And then once that is kind of, it's held, right, by the peptidyl transferase actually in the large subunit, and then we have the peptide bond formation form between the carboxyl end of the methionine and the amine end of the serine, right? And that's in a hydrolysis, not hydrolysis, a condensation reaction, condensation dehydration reaction. And so once that happens, now the original tRNA is discharged or uncharged, and both of the amino acids are now attached to the second tRNA. So you can just see this is shown in an actual kind of more detail, right? Showing you where the water comes from and where the peptide bond is formed between the two. Come on. Okay, so now that we have this, now we need to move the whole complex along by one codon. And that's carried out again with the use of GTP, or the energy in GTP, and a second elongation factor called EFG, right? And again, that's its analogous one is EF2 in eukaryotes. And so that comes in, actually binds to the small subunit, the bit which is in contact with the mRNA. Just find that bit. Where are we? There we go. Right. And the hydrolysis of that phosphate bond in GTP provides the energy to move the whole process along, called translocation. And so once that is done, there we are. We now have our discharged amino acid, tRNA, sorry, in the E site. We have our second tRNA now with the 2 amino acids in the P site, and our A site is open. And so this tRNA needs to leave, and another tRNA will come in that's already charged with EFTU and GTP, and the whole process continues, which you can see here. We've got another one. This is for lysine because its anticodon UUC recognizes the codon AAG, right? Our methionine tRNA has already left, and then we begin the whole process all over again, right? These two are then cut off from the second, the serine tRNA. Peptide bonds form between serine and lysine. EFG and GTP comes in, translocates along, and over and over and over and over again. And that's essentially how translation works. Does anybody have any questions about that stuff? So it's basically a 3-step process. tRNA comes in with an amino acid. Existing amino acid or amino acids, right, the polypeptide chain, if it's been going for a while, gets transferred onto that new amino acid. Whole thing moves along with the aid of GTP. tRNA leaves, another one comes in, over and over and over again. And this can happen, some depends on the species, but some proteins can include thousands of amino acids in just one kind of intact protein. And as you can imagine, because so much, well, essentially 2 GTP are required per amino acid that's added, right? You can see how to make one single protein of, say, 1,000 amino acids, you're going to need 2,000 GTP, which is astonishing amount of energy, right? This is why gene expression, or one of the reasons why gene expression is so energy intensive, right? It's not something that you do willy-nilly, right? Everything has to serve a purpose. So when we think about the control of gene expression, obviously you want to turn on the right things at the right time because that's important, right? So you can do whatever function that cell needs to do. But also you don't want this happening unnecessarily because it takes up a lot of energy. And so, you know, you'd be at a selective disadvantage if you just wasted the energy you got from your metabolism on unnecessary protein synthesis. And that's besides all of the energy required in obtaining or making these amino acids, charging the tRNAs with amino acid transferase, which is back up here, which also uses energy in the form of ATP, right? And so all of this takes a lot of energy. Okay. Wait for, and I'd strongly recommend watching that video. It's really quite simple, but it lays out all the steps in a very straightforward fashion. It's a very nice, clear video. Okay, just giving people time to think. Also clean my glasses so I can see what I'm doing. Also have doggo number 2 on the other couch, looking kind of forlorn. Okay, so if no one has any questions, I'll move on to termination. And essentially the deal is here you have 3 potential stop codons. And the genetic code is the same both in prokaryotes and eukaryotes. It's a very ancient, evolutionary speaking, deal, essentially. And just as you have a start codon, right, that's methionine, you have 3 stop codons, UAA, UGA, and UAG, right? And these do not have a tRNA that recognizes them, right? So when a ribosome encounters one of these in the A site, translation stops. And so essentially there's 2 parts to this. One is that translation stops and it can no longer proceed because there's no tRNA to come in, but also that kind of paused ribosome and that stop codon are recognized by something called release factor. And so the release factor's job is to promote the final hydrolysis of the bond between the last amino acid and the last tRNA in the P site, and also to dissociate the ribosome from the mRNA, right? And so the release factor promotes both of those, also with the aid of GTP again, right? So GTP is the key energy component for protein synthesis, at least the actual turning of the mRNA into a polypeptide sequence. Obviously, ATP is also used for charging the tRNAs as well. So the ribosome translocates over one more, and there's an stop codon in the A site. As soon as that's there, the release factor comes in. Hydrolysis of the bond between the last tRNA and the last amino acid. And then the dissociation of everything else, the large subunit and small subunit, and the tRNAs that were present also leave to go on and be recharged by the amino acyl synthetases. Right, and that's pretty much it, to be honest. Right, so that's the core of translation. And so really what I want to do now is talk about some of the kind of, you know, tangential stuff, essentially. So one of the things which is super important, so I think someone might have their, Alejandro, I think you have your mic. There you go, thank you. One of the neat things though is that this doesn't happen one ribosome at a time, right? So whenever you produce a mRNA, you're now able to translate it with multiple ribosomes at the same time. And these are called essentially polysomes, right? So basically ribosomes are huge, right? And so they're very large protein complexes of tens of proteins and at least 4 different ribosomal RNAs that I can think of, right? And so these occupy a lot of space. And basically they occupy a space corresponding to about 25 amino acids. So if you think about that, that's about 75 base pairs of an mRNA. But once that ribosome complex is moved off of the AUG, of the start codon, now another translation or another ribosome can assemble around that start codon, whether it's directly in prokaryotes or if it's indirectly through the scanning mechanism in eukaryotes. Once that start codon is free, another ribosome can begin the process of translation of that mRNA at the same time, essentially. Right, and so when you have multiples of those, essentially you get what's called the polysome. And so this is a very efficient way of ramping up production of a protein from an mRNA. Because all this takes time, right? It takes time to turn on the gene, takes time to transcribe it, process the mRNA. That mRNA has to get out to the cytoplasm. And once, so this takes time, even if it happens in, a relatively short span of time, at a cellular level, it still takes time. And so once that mRNA gets out there, now you want to make the most of it and make as much protein as you can, right? Particularly if that's a protein that you need kind of like right now. And so a polysome is a very effective way of doing that. So there are also some kind of funky differences in translation in prokaryotes and translation in eukaryotes. So in eukaryotes, I don't think we have, oh yeah, we do. Where is it at? Well, this will do. Essentially, each gene is its own unit. It's not completely rare for genes to be clustered together, but each gene is transcribed independently in eukaryotes, right? So it's one gene, one mRNA, essentially. In prokaryotes, we have something slightly different. So now we have something called cistrons. And a cistron is basically a stretch of DNA that has multiple polypeptide encoding bits in it. So we would normally call these a gene, right? In eukaryotes at least, one gene equals one mRNA equals one protein. In prokaryotes, actually what we now have is one gene equals one mRNA equals multiple proteins. And this is called a cistron, right? And these transcripts are polycistronic, right? So essentially that means that you can produce lots of different proteins from one single mRNA. Now, these individual proteins have their own start and stop codons. It's not produced as one giant protein that's then kind of sliced and diced. And so the information sets are distinct on each one of these mRNAs, but they're all part of the same mRNA. And so one of the reasons for that is that in bacteria, genes that are involved in the same process are almost always in a cistron. Almost always. So for example, in metabolism, right? If you need to produce a series of enzymes or transport proteins or whatever, right, involved in the metabolism of a particular substrate to produce something else, all of the genes that are involved in that metabolic pathway are grouped together. So essentially, if you go, oh, shoot, look at this, I've got some galactose in my diet now, if bacteria could talk to each other or themselves even. Essentially, you go, okay, well, I've got this new food source. I'm going to need everything, right? So it's not an a la carte menu. It's like a program menu. What do they call that? I don't know. I haven't been to a restaurant for so long. I can't remember what they call the opposite of an a la carte menu. But anyway, essentially, you go, okay, I want that. And so you get the whole caboodle all together. You get proteins involved in import of that substrate, proteins involved in the metabolism of that substrate, and whatever else is needed tends to be in one large instruction set, right? Where the coding for the import protein and the metabolic proteins are all present as distinct bits of information in one large mRNA. And so it makes life a lot simpler in many ways, right? Because in eukaryotes, we have the same metabolic pathways to a greater or lesser extent, but now we have to coordinate expression of all of those from individual genes, which may be scattered throughout the genome, different chromosomes. Whereas prokaryotes have a much more streamlined process where everything is grouped together in one single kind of unit of information, essentially, one cistron. And so a lot of how prokaryotes control gene expression is tied to the control of expression of a single cistron. And so you'll have basically all the components required for controlling expression of that cistron at the beginning of that gene. So that's one particular oddity between prokaryotes and eukaryotes. Another really, really big one, and this is super important in terms of control of gene expression in prokaryotes. It's actually one of the main ways, or yeah, not one of the main ways, one of the funkier ways, let's put it that way, in which prokaryotes control their expression of their genes is that because these two things do not have to happen in different compartments, in eukaryotes we have transcription in the nucleus and translation in the cytoplasm or the rough endoplasmic reticulum. In prokaryotes, it all happens in the same space. So as soon as you start producing the mRNA, as soon as enough of the mRNA has been produced for that Shine-Dalgarno sequence to, become available, translation begins. Even before that mRNA has been finished being transcribed, right? So we call this the coupling of transcription and translation. So those two processes occur at the same time. As soon as enough of the transcript has been made, ribosomes assemble, and you have these polysomes forming before transcription has even finished, down there at the end of that cistron, which I think is pretty wild. And that allows prokaryotes to use features in the mRNA as a means to control translation. And we'll get to that. That's the kind of canonical, or the poster child of that is tryptophan, control of the trip operon. Oh yeah, and probably the better term for this collection of essentially kind of genes, I guess, is called an operon. Let's just bring that up. And those operons produce those polycistronic mRNAs. Actually had a friend this is going back many, many years, when I was an undergraduate at the University of Birmingham in the UK, who was a DJ, and his handle was DJ Operon, which is pretty good. Anyway, okay, so and that's definitely not the case in eukaryotes, right? We cannot do this in eukaryotes because these things occur in different compartments. And so just to finish up, essentially what I want to do now is talk a little bit about the genetic code and essentially the consequences of mutations, and also kind of one of the things that we can do to guard against that, or one of the mechanisms that we have, right? So essentially, as you know this already, right, genetic code consists of all the codons and the amino acids that they encode, right? And this code is degenerate. And so what we mean by degenerate is that more than one tRNA or more than one codon can produce the same amino acid, right? Not that it has like low morals or something. So more than one tRNA can, let's put it this way, more than one codon can encode the same amino acid. But, not bit, but only one, basically one codon, come on, equals one tRNA equals one amino acid. Aha, gotcha, I think. And so that's really important to stress because here we go to the genetic code here. So this is a codon table, which I'll explain in a little bit. So even though, you know, at least, there we go, two codons can encode, what's that, aspartic acid, ASP. So if you have GAU or GAC, you will get the same amino acid in that position, which is aspartic acid. But the tRNA that recognizes GAU will only ever have aspartic acid added to it. And it doesn't do a very good job, well, recognize almost solely GAU. It can kind of recognize sometimes GAC, but it's very specific to that codon. So even though, and there's some others in here, which is like, which is the big one? Leucine, there you go. So leucine is encoded potentially by 6 different codons. But the codon, the tRNA that carries leucine and has an anticodon of GAC, which recognizes as CUG, is not going to enter the ribosome and bind when you have a UUA or UUG in the A site, right? So even though you can get the same amino acid through many different codons, and it varies from, you know, 1 to 2 to 6, those tRNAs are still very specific in terms of the anticodon, the codon they recognize, and the amino acid that they carry with them, right? So it kind of goes one way. You can have multiple ways to get to the same amino acid. Doesn't go the other way. Each tRNA cannot have more than one amino acid. It only ever has one, and only ever recognizes one codon, with a few exceptions. And so this also, if you think about it, provides a degree of buffer, right? And so essentially, if you have a point mutation, like so you change, let's say you have CCG in your DNA and you change that to CCA, or it changes, not that you change it, right? Random change turns a G to an A in the third base of that codon. The result, nothing. So CCG and CCA both encode or are recognized by tRNAs that carry proline. And so even though you have a mutation that has occurred, right, there is a base difference between those two sequences. The amino acid sequence does not change. So if you think about it in terms of just kind of like numbers, right, if you have an open reading frame, essentially, even if mutations occur by random chance within that open reading frame, as long as they're conversions, right, from one base to another, you don't have, there's a high chance that you're not going to have any change in the amino acid sequence. Essentially, you've got a 33% or perhaps a bit less, perhaps about 25 to 30% chance that no change will occur as a result. And so even though we often think of like mutation as the thing that drives novelty, right? It's how we ended up how we are. It's this collection of mutations and other funky stuff, basically, that occurred over our evolutionary history. Generally speaking, mutation is a bad thing, right? You know, we always think of, let's go into a, what was it that turned Bruce Banner into the Green Hulk? I don't know, it kind of depends on which version you watch, right? But basically it's like some massive burst of radiation that this scientist absorbed, right? And we always think, oh yeah, it kind of ends up with, you having superhuman strength and turning green and stuff and being angry all the time. But it doesn't actually. It generally tends to kill you, right? Because it damages DNA and you need that information to do general daily life type stuff at a cellular level, right? So almost mutation is always generally either bad for you or bad for the gene or has no effect on the gene, right? It's very rare that a mutation actually improves function or generates a novel function. And it's also really, really worth pointing out this whole deal of the reading frame. So if you remember way back here, right, this start codon is what determines the frame, that series of 3 letters that is used by the ribosome to produce the amino acid sequence. And so that frame starts with the start codon. That's how it is determined. And essentially, from that point onwards until you get to the stop codon, that frame exists in a series of 3 letters. Codon, 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 codon. There's nothing in this sequence which tells the ribosome, other than the start codon, what that frame is. All the ribosome will do is move 3 bases along to help with GTP, add another tRNA and amino acid, move 3 bases along, add another tRNA and amino acid, right? It doesn't know whether it's in the right frame or not. And so the types of mutations which tend to be really damaging are ones which are called frame shift mutations. So if you take out or add bases, particularly 1 or 2, you can really, really screw things up. And this is kind of like a kind of exceptionally convoluted way of kind of going through that. But essentially, the ones that I think are particularly useful to look at is the first one, right? So this is the frame. And I don't know, whoever made this found a lot of 3-letter words, right? Because that's what helps. But essentially, the frame is determined by the, right? So this would be analogous to the start codon AUG. And then every single word comes in units of 3 letters after that. So if you add a letter, let's say you add another T here, the ribosome is not going to know that it should skip that T or U in the mRNA. Right? All it will see is, okay, well, this is the word. Bring in the amino acid for this particular word. even though that word is now wrong, right? And so if you add a single base, you essentially push the frame back by a letter. And so you can see the existing one is here, right? The new cap, whatever it is, eat the hot dog or whatever, right? But now, once you've added that base, okay, we now have another 3 letters. Now it's TTH. The next 3 letters, the next word is ENE. The next word is WCA, right? And so this has, and you get the same when you take out a letter too, right? This would be a single base deletion, which you can see. Oh, yes, you're very cute. You can see down here. So that has 2 consequences. One, you obviously get gibberish afterwards, right? So these new words encode different amino acids. So you'll get part of the correct amino acid sequence at the beginning until you get to that insertion or deletion. And then after that, it will be something completely different. So that's one thing. So that's bad enough in and of itself, right? That would generate a protein which has a completely different structure, and who knows what it would do. Most likely it would just be a kind of clump of non-functional protein, which has its own issues as well. That's actually part of the reason behind Parkinson's, if I'm right. Yeah, I think so. So in Parkinson's, you get a large clumps of proteins called aggregates forming, which after a certain level, your neurons can't cope with that and they die. So even having this kind of gibberish protein around is not going to do you any good. But the second consequence is that this new frame, right, is not normally translated, right? And so it may have in here, in fact, almost certainly will have at some point, a stop codon. And so typically the result that comes from an insertion or deletion is a truncated protein, where you have your original protein sequence, a stretch of gibberish, and then a stop codon. And so these almost always, in fact, like 99.99999% of the time, generate a non-functional protein. Occasionally, if you're, it's kind of funky, right? Where's a +3? Yeah, I don't think there is a +3. If you add or take out 3 letters at a time, the frame is maintained, right? So you don't have that new stop codon popping up. You don't have gibberish, but you lose or gain an amino acid, which may or may not have a big effect depending what that amino acid does and what one has been added. So here's a little bit of a kind of a twist on the 1 tRNA equals 1 codon. That's not always the case, right? So for some of these, and you'll find that it's really for ones where the third base, right, the basis of that degeneracy is variable, but you get the same amino acid, you can have something called wobble, right? And so basically that means that the first 2 bases of the anticodon are the ones that really, really matter. And so essentially, and the third one kind of does, but there's a lot less stringency about it. And so essentially what you have is actually there are a few codons, that will recognize all of these 4, a limited number of tRNAs, sorry, that recognize these 4 codons. But because they have the same amino acid, it doesn't actually matter, right? So there's a little bit of, when I said, 1 tRNA, 1 codon, in theory, that's true, but in practice, there's actually fewer tRNAs than there are codons. And so the specificity is a little bit less than what I've been laying on. But the same principle kind of applies. Oh, sorry. Another puppy shot. Isn't she cute? She's heading off to Ohio on Thursday. She's been adopted by an older couple who have another couple of rescue dogs. So a little caveat there is, well, kinda, right? And so basically, codons that share the same first 2 bases and result in the same amino acid being added can be recognized by the same, can be recognized. So that's a little bit harder to explain, right? Because it's not as absolute, but the same principle kind of applies, I guess. Hopefully that makes sense. And the reason for that is because there's, what is that? How many codons are there? Different codons are there? 64, I think. There's a lot. And so rather than having that many individual tRNAs and amino acid synthetases, well, we kind of fudge things a little bit and we use fewer, but we end up essentially with the same outcome where these codons equals this amino acid. Those 2 codons equals that amino acid, right? So essentially that pretty much wraps it up for gene expression, transcription, and translation. And so I don't have anything else to cover. So what we're going to do on Wednesday is use that session as a recap question and answer session. I'm not going to go over all of the material because it's taken us, what, 3 weeks or so to cover this much material. I'm not going to be able to do that in 50 minutes. It would be wild. So what I want you to do instead is, those that are present, which isn't very many of you, but I'm glad you're here nonetheless, is go through your notes, go through all the PowerPoints, try and quiz yourself about stuff, right? And that's the key part. And figure out what you don't know or what things don't make sense, right? Don't just go through the PowerPoints and like stare at them and go, oh yeah, that will make sense. Because there's a good chance that you may think it does, but it actually doesn't. So try and figure out what doesn't make sense, what things you need help with, and bring a list of those with you on Wednesday. And it will be nothing but you asking me questions and me answering them. So if you don't ask me any questions, it'll be a very short lecture. But if you do, then we'll just keep going for the whole 50 minutes until hopefully we've answered all of the questions that you have. And your understanding is, that much better than when you started. Because the exam is on Friday, right? And it will be it'll be a tough one, right? I mean, they all are. But it's going to be lots of details, lots of how does this work? How does that work? Why does this work in a particular way? What are the differences between this, that, and the other, right? And so it really will pay to get a good grip on this, and that will help you be successful. And so Wednesday is going to be one way of me helping you do that, right? But you also have to help me by bringing a bunch of questions with you. Otherwise, I'm just going to be like, I'm going to be sitting here. I'm going to be sitting in my office, drinking a cup of coffee, staring at the screen, right, which would be sad. So make a list of questions, anything that you want to ask about the material we've covered, and we'll go over that on Wednesday. What PowerPoints? It will cover. Where are we at? So, where are we at? I've got to look up the. So basically, PowerPoints 12, 13, 15, and 16. So genome structure, transposons, transcription and translation, and then RNA processing, obviously. So 4 of them in total, I think. Yeah, that's right. No worries. Okay, everybody. Thanks very much. I shall see you on Wednesday. Take care and study hard.